Reversible cell injury. If the ischemia or hypoxia is of short duration, the effects may be reversible on rapid restoration of circulation. Example in coronary artery occlusion, myocardial contractility, metabolism and ultrastructure are quickly reversed if the circulation is restored. The sequential biochemical and ultrastructural changes in reversible cell injury are as under. First, there is a decreased generation of cellular ATP, damage by ischemia from interruption versus hypoxia from other causes. All living cells require continuous supply of oxygen to produce ATP, which is essentially required for a variety of cellular functions. Example, membrane transport, protein synthesis, lipid synthesis, phospholipid metabolism. ATP in human cell is derived from two sources. Firstly, by aerobic respiration or oxidative phosphorylation, which requires oxygen in the mitochondria. Secondly, cells may subsequently switch over to anaerobic glycolytic oxidation to maintain a constant supply of ATP, in which the ATP is generated from glucose, glycogen in absence of oxygen. Ischemia due to interruption in blood supply as well as hypoxia from other causes limit the supply of oxygen to the cells, thus causing a decreased ATP generation from ADP. In ischemia from interruptions of blood supply, aerobic respiration as well as glucose availability are both compromised, resulting in a more severe and faster effects of cell injury. Ischemic cell injury also causes an accumulation of metabolic waste products in the cell. On the other hand, in hypoxia from other causes like from those of higher RBC disorders, heart diseases, lung diseases, anaerobic glycolytic ATP generation continues and thus cell injury is less severe. However, highly specialized cells like those of the myocardium, proximal tubular cells of the kidneys and neurons of the CNS are dependent solely on aerobic respiration for their ATP generation, and thus these cells suffer from the ill effects of ischemia more severely and rapidly. Intracellular lactic acidosis. Nuclear clumping due to low oxygen supply of the oxygen to the cell. Aerobic respiration by mitochondria fails first. This is followed by a switch to anaerobic glycolytic pathway for the requirement of ATP, that is ATP. This results in a rapid depletion of glycogen and accumulation of lactic acid, lowering the intracellular pH. Third, damage to the plasma membrane pumps, hygropic swelling and other membrane changes. Lack of ATP interferes in the generation of phospholipids from the cellular fatty acids, which are required for the continuous supply of membrane repairs. This results in the damage to the membrane pumps operating for the regulation of sodium and potassium, and along with this calcium. Fourth, reduced protein synthesis. Dispersed ribosomes. As a result of continued hypoxia, membranes of endoplasmic reticulum and Golgi apparatus swell up. Ribosomes are detached. The rough endoplasmic reticulum and the polysomes are degraded to monosomes, thus dispersing ribosomes in the cytoplasm and inactivating their function. Similarly, reduced protein synthesis occurs in the Golgi apparatus. Ultrastructural evidence of reversible cell injury damage is seen in the form of loss of microvilli, intramembranous particles and focal projection of the cytoplasm, bleeps. Myelin figures may be seen lying in the cytoplasm or present outside the cell. Up to this point, the withdrawal of acute stress can reverse the changes of reversible cell injury.